And what we're doing this week is, well, 1 week from today, we have an exam in this room. Remember, I'll put out some identifying name tag type things so we get everybody spread out in here. And it's going to be 50 2-point questions like that. Sometimes they get bundled in together. So it's a total of 4 points because it's like give 4 examples like that, and I just like fit it together like that. And remember the concept that you're just going to have to keep moving because I've taught this course a bunch and I know how to write an exam that fills up the time, so you won't have extra time. You just have to keep working along on the exam. And we've got more stuff to talk about. Our receptors in channel 16. The chapters are 1, 7, and 16, and everything we've done. And then we also have an assignment today, and it's a pretty long video. I think it's a 47-minute long video to fit all that stuff together. And with this, all of our courses are always kind of leaning ahead, looking ahead, getting set up to go further ahead. And after the exam, we'll cover chapter 17, that's the cell cycle. Okay, so that's where we're at on that. And there was another comment I had. So our first exam is going to be on the 24th. We have a slight change. I updated the syllabus. Slight change on when our second and third exams are going to be. And yes, Chase.
Did you say chapter 17 is after exam 1?
Correct. So what's going to be on exam 1? Chapter 1, chapter 7, chapter 16. Chapter 16 is about the receptors and signal transduction pathways that we've been learning about. Chapter 17 is about the cell cycle. In the PDGF receptor video, though, I talk about how it's working under our scab to get our skin cells to go through the cell cycle faster. And so whatever we talk about in the cell cycle in that video will be on this exam. But you certainly don't have to look at chapter 17. Just be ready to answer everything that we've covered. And I just moved the exam 2. I moved back one class period. It was on a Tuesday. Now it's on a Thursday. Exam 3, I moved it forward. One class period, it was on a Tuesday, now it's on a Thursday. And it's just, I don't need to explain, the details of why, I did that. But it was just a slight move. And like I said, it's updated on the syllabus, and of course, we'll be talking about that. So there is, a big focus of the PGF receptor assignment, and putting it in the context of wound healing. It works in a lot of different settings, but that's a really, really good one to always remember. And and we learned a lot about that specific pathway. And here's one more fact about that pathway. So we need to know that it's PDGF, then it binds to PGF receptor, then that binds to that linker protein that we don't need to know of to activate RAS. And then RAS, as long as RAS is bound to GTP, it's going to be activating RAF. And then RAF is a kinase that activates MEK. And then MEK is a kinase that activates ERK. And ERK is a kinase that activates ELK. And when phosphorylated ELK binds to an enhancer of genes that can include MEK, and we'll learn of several other genes that will go back to the cis-regulatory region of ELK and add more enhancers and more transcription factors after the first exam. But for the first exam, we've learned that, that enhancer that binds to ELK that would be in the cis-regulatory region of genes like MYC. So ELK target genes includes also this SRF, and we learned good stuff about that. And just to add this term, to add just some more awareness of this, and these are often referred to as the MAP kinases. And we know why they're in a chain like that. They are going to amplify the signal. One enzyme can go coo with a high turnover number. And so one RAF can activate lots of MEK, and then one MEK can activate lots of one ERK, and so forth like that, okay? And I think I have one more slide of that. So in fact, in chapter 16, it shows that RAF, MEK, ERK are actually often bound together, held together, just so they can do a kind of a bucket brigade amplification. And this is called a scaffold protein. And they're holding those, and it's because they were discovered really at the same time, and they're doing the same thing, and sometimes they're in a scaffold, bonding situation. People refer to them as MAP kinases, and we can learn that term also. And it's just those three, and there would be other proteins that are also MAP kinases, but these are the really famous ones. And what that stands for is a good term for us to know. MAP stands for mitogen activated protein kinases. That's a good term because this is a nice term for us to know. Mitosis, so it's going to activate the mitotic cell cycle, and so mitosis generating. And so, you know, super keen on that. PDGF is a mitogen. It stimulates the mitotic cell cycle. And we will see other signaling factors that are mitogens. In that, that serum, in that fetal calf serum that people have to add to the tissue culture cells, to add to just the nutrients, you know, they need a stimulator, a mitogen that's in there like that, okay? So just a little term just to kind of keep adding to how much we know about that. Okay, great. And we learned a lot about that, and then we learned about that there are other signal transduction pathways that 58 members of the human tyrosine kinase receptor family, gene family, it can activate. And there are more than this, but you know, let's just dive into a little bit of that to be aware of that. So we got the PI3 kinase story and that phospholipase C story. We'll just add a little bit to that. So in that context, remember, well, these are all receptor tyrosine kinases, so they fit into this gene family, and this gene family does the same thing, like that. They all, when bound to their specific ligand, they all dimerize, and then they autophosphorylate to activate them, and they all activate signaling pathways that can include RAS, and we know a lot about RAS, like that. And, or they can activate this PI3 kinase pathway. We saw that. That's our first slide, first day of the class, where we're going to activate this FOXO by phosphorylating, especially this AKT. And AKT is a very famous kinase, and it directly phosphorylates FOXO and causes FOXO to be inactivated by sequestering it in the cytoplasm. This page that's no longer in the textbook, they just were streamlining things like that, you know. So we have our third example, and it's this phospholipase C, and that's the one that clips these into DAG and IP3, and DAG is going to go on to bind in the membrane to bind and activate protein kinase C. So down a pathway, and that's all we need to learn about that pathway. Is it's just a pathway. And then IP3, I just want to mention a little bit more about that. Just a couple word terms. So that's the one that can float around inside the cell, in the cytoplasm, and it can bind to receptors in the membranes of organelles, like this IP3 receptor, and that can cause a release of calcium, like that. So an increase of calcium, and calcium is a second messenger that can bring changes into the cytoplasm that leads to, you know, this variety of cellular responses. And the two terms we want to add to is just to say that those are things we just talked about. Where's the words? Is that IP3 receptor, we have a better name for that. It's called the ryanodine receptor. That's the receptor for this IP3 signal that comes from that phospholipase C pathway. So this is that third pathway you're learning about, where phospholipase C clips IP2 into DAG and IP3. Then IP3 binds to an internal IP3 receptor that's called the ryanodine receptor. And then the last term I just want to show you is this one, and that is that calcium, that increased calcium in the cell. Right, that calcium ion it's a second messenger, and a very, very common ortholog in animals is a protein called calmodulin. And that's kind of a nice name because calmodulin is bound by calcium, those calcium ions, and then that becomes a signal. So it's an indicator of increased calcium concentrations. We get activation of calmodulin, and because it's a protein, it can bind to lots of different proteins and other molecules inside the cell and regulate them. So it's not just, oh, it's that calcium and that's all you need to know about that. I don't want to say most of the time, lots and lots of time when we talk about increased calcium, what we're really doing is activating this really, people say ubiquitous. It's just every cell has calmodulin in it, and calmodulin's function is to detect increased concentrations of calcium and activate lots of cellular responses. Lots of these are non-genomic responses. They're happening in the cytoplasm. Things that would be changes in metabolism and changes in the cytoskeleton and things like that, okay? So just a little bit more to add to what we learned about that, okay? And so that's the receptor tyrosine kinases, right? And we've learned a lot about that. We want to add it to what the book has and what the book might add. And then the book actually talks first and somewhat more about G protein coupled receptors. And so we learned base knowledge about that. And then last week, last Thursday, we talked about 2 examples of signal transduction pathways that G protein coupled receptors use. And they're really good examples because the first one is about what happens when we have a fight or flight response and we actually get a sugar rush inside our muscle cells, not from sugar being picked up, but sugar just being produced by breaking down glycogen. And so once again, we want to know the basic story of G protein coupled receptors and how they bind to a trimeric G protein. That's why they're called G protein coupled receptors, and activate signal transduction pathways like this one. There's the name I forgot. Glycogen phosphorylase is the enzyme that cuts the glycogen into glucose. And I forgot it. I always forget it because it doesn't cut it into glucose. It cuts it into you can call it glucose. Everyone would just say glucose, but it's actually a phosphorylated form of glucose. So that right? Okay. So the activated G protein, the trimeric G alpha subunit, binds to adenylyl cyclase, and that's an enzyme that catalyzes the conversion of ATP to this second messenger cyclic AMP, which binds to protein kinase A, activates it. It activates another kinase to amplify, and it activates that enzyme that just directly breaks down glycogen and gives the muscle a sugar rush. Okay, and then we talked about this just amazing story that's related to addictive behaviors. The dopamine receptor, there are 2 types. There's a non-cellular response that dopamine is a neurotransmitter, elicits in neurons, and that is it opens channels. It causes the action potential, the sort of electrical signal to go from one neuron to the next. That's all the time we think and behave. We're always having those events occur, and dopamine is one of those. And then in addition to binding to that receptor, it binds to this other receptor that's a G protein coupled receptor that activates a genomic response. And so, again, not a course in neurobiology, but it is good for us to be aware that, all that neurobiology is one neuron talking to another across the gap called a synapse using molecules called neurotransmitters, and dopamine is a neurotransmitter. We would call these signaling molecules. It's just generally the case. They would be called paracrine signaling factors, but let's call them neurotransmitters. And the specialized areas of biology, neurobiology, immunology, developmental biology, they all have different general terms. Neurotransmitter and neurobiology, the kind of comparable factors, little signaling factors that bind to receptors and target cells in the immune system are called cytokines and chemokines, and so we're at neurotransmitters. So the neurotransmitter diffuses across here, binds to these receptors. It's showing the channel receptor. It's not showing the other receptor, which is the G protein coupled receptor that activates not just the non-genomic response, but also this genomic response. And the genomic response is nice because it's that same pathway through G protein coupled receptor onto the G protein and adenylyl cyclase. Phosphorylated cyclic AMP binds down here and phosphorylates CREB, nice name for it. We talked about that cyclic AMP response element binding protein. So really nice learnable stuff in there. And then that's the last slide we ended up last time. So I'll stop there because I'll say, I'm done. We can answer lots of questions, but I'm done, with what I wanted to say about those 2 major classes of receptors. and to be open questions about anything at all. And it, you know, it can always be from any part of our course. You can still ask questions about the syllabus or my office hours or anything like that, all the way down through. And you know, it's my job to put your question in context of where we're at. So don't worry if you're like, well, you know, a lot of times students will say, I don't think, you know, this is the right place, or, you know, I just have one that's a really different kind of question. Just ask it, right? Right, like, you know, you're my boss, right? You're paying my salary, like that. One time I calculated, like, you know, like, if you add up all the tuition, and then you know, divide by the class number and stuff, I figured out what the cover charge was for you to come in for one class meeting, you know? You should calculate that sometime, right? Like you're going out, or like going out to a show, and you gotta buy a ticket for just not the whole semester, just that one class meeting, like that. The cover charge is pretty high, right? They just come in here and sit around and be like, well, that was fun. Yeah. So, yeah, we should ask questions, right? You should get your money's worth out of it. But yeah, anybody have any questions right now? So I have this class, well, Thursday, and you know, we'll get through everything, and if we do, I'll just start talking about stuff that's beyond, you know, our exam. That'll be on second exam, and that just gives us plenty of room for, you know, questions and review and that type of stuff. Okay, and it could be from your textbook, right? I always bring my textbook over. So you can see what we have here is we've talked about nuclear receptors, the receptor tyrosine kinases, G protein coupled receptors, and we want to talk about these other two groups, and our book covers them. And the next one is called non-receptor tyrosine kinases. We'll talk about one example. There are 35 of them in our body. They're not really closely related. They are, so that's where we're at. And the one we're gonna learn about is called JAK-STAT, and it is in our book. And it's a very famous one, and that's one reason these tags are down here is they don't fit into, you know, big groups of receptors, but you know, they're very, very important receptors. And so here's JAK-STAT, and it's just a great one. So JAK is the receptor, and STAT are transcription factors that get activated. So that's the signal transduction pathway in the name. We're saying the name of the receptor, JAK, and the major target transcription factor is called STAT. And so here's a non-receptor tyrosine kinase, and all it means is that it's a receptor, and the tyrosine kinase enzymatic activity is not part of the receptor, it's part of another protein that's bound to the receptor. You know, so it just puts it in a different category like that. And so this non-receptor tyrosine kinase, this sort of pink or orange blob, that's JAK for us. And there are 35 different examples of these in humans, right? Like, so there are several of these, and they have that story. There's a receptor, and we don't even talk about the receptors have names, but we almost always talk about, well, what's that kinase that doing, and you know, what's it gonna do like that. And so you see it does the same kind of thing. It's gonna autophosphorylate when the ligand is bound to the receptor, and then here's what it does. It autophosphorylates, and then it phosphorylates this target molecule called STAT. And when STAT is phosphorylated, it's an activated transcription factor. So super straightforward. Look what we do. This is a big part of our first exam. We have lots of big parts to our first exam. Phosphorylated by kinase examples. FOXO gets turned off when phosphorylated. ELK gets turned on. STATs get turned on. And that's it for that one. Non-tyrosine receptor kinases, okay? All right, and then this next one is very unique. They aren't in a group, there's only one. They sometimes are in a gene family, like a small gene family, but it's really just just their own kind of category like that, right? And you can think of that, you know, in other kind of stories, right? You can group some of your friends together in groups like that. Oh, these people all play hockey, but there is a friend who's unique, and it's just like that. It's just, you know, these are kind of somewhat arbitrary, not really arbitrary categories, but it's how all the scientists talk about these cell membrane these cell receptors. Yeah. So the first one we're going to talk about, people usually call it the Wnt signaling pathway, and Wnt is the ligand, the signaling molecule. And here's the pathway. It's in our book, and it just shows in the absence and the presence of that Wnt signal. And so here's Wnt. It's going to be that extracellular signaling molecule. It's going to bind to the receptor. The receptor is called frizzled, and frizzled is going to activate a signal transduction pathway that in this case leads to the stabilization of a transcription factor called beta-catenin. And it's in your book, and I know it'd be nice for you to see all those, but I can just write them down here too. So Wnt is the signaling molecule, frizzled is the receptor, and then especially important is this beta-catenin. And beta-catenin is a transcription factor, and in the presence of Wnt, the beta-catenin is stable, and when there's no Wnt, beta-catenin gets degraded. So that's a new kind of transcription factor for us. A new kind of regulation is it's either stable or it just gets destroyed. And weirdly, it's just destroyed almost all the time, right? It's just constantly being made and getting destroyed because there's no Wnt. And then Wnt comes, signal, stabilizes it, does its job. What's its job is it goes into the nucleus and it binds to a transcription factor. So here's the DNA. And there's already a transcription factor found in DNA target genes called TCF. And so TCF has a DNA binding domain and a transacting domain, and TCF is a big time repressor. So in the absence of Wnt, beta-catenin gets degraded, and TCF gets to do its job, and its job is turning genes off, right? When Wnt shows up, that pathway gets activated through frizzled and the steps in that signal transduction pathway. Beta-catenin binds on the back of TCF, so beta-catenin is an example of a co-regulator. Beta-catenin does not have a DNA binding domain of its own. It takes advantage of that one, and the beta-catenin activates. So we have a really nice example of a molecular switch like that. These genes are off. Now beta-catenin and the Wnt signaling pathway turn them on. And so that's just a beautiful molecular switch, off and on. A crisp switch, like if you had a light switch, and it was floppy, it would sometimes just kind of flop down, it wasn't really crisp. And this is like somebody's holding, TCF is holding the light switch off. There's a finger called TCF that's holding it down. And then beta-catenin shows up, and it's a different finger, and it lifts it up. Even though TCF continues trying to hold it down, we see the beta-catenin is stronger. And it turns out that scientists really enjoy discovering those because, in chemistry, there's always different concentrations, right? The concentration's going up, and that's a gradient. It's not a threshold like that, but we often want to have on-off switches, and we saw that where the nuclear receptors are big-time switches. And it's curious to see, curious to kind of face this reality that it looks like a good molecular switch. And then the reality is, yeah, so it is, and it's really, really ancient, and it's used over and over again. So this beta-catenin and the Wnt signaling pathway is found in every animal on our planet, and every animal on our planet, and no plants. Very early in the evolution of multicellular animals, this pathway was invented, and then there are literally easily identified orthologs of every protein in the pathway in every animal on our planet. So if you said, why is this odd signaling pathway in our book? Well, kind of for us, it's especially in there because we have a totally new example of a transcription factor that's got this molecular switch, and the molecular switch is it gets degraded. It just gets destroyed, and then stabilize to switch it on and off. And it's the Wnt signaling pathway. And remember, there's an amplification in that, right? So the degradation is another kind of layer of it's off, it's really off. And then you get this massive amplification with the presence of a little bit of Wnt. So a massive switch like that. And it, that's probably why it's seen universally in animal bodies, like that, animal cells, signaling, like the simplest little jellyfish. There's none of it in sponges. Some people think of sponges as animals. But in Nigeria, which are like jellyfish and hydra, they have orthologs, not just of Wnt, but also frizzled, the receptor, and then disheveled, and GSK, and these the pathway stabilized beta-catenin, and beta-catenin, TCF, like that. Just orthologs of everything in the pathway. So that somebody would say, who's working on it, would say, with regard to the overall significance of life on Earth, beta-catenin is equal to everything else in that chapter. That Wnt signaling pathway is just used. And you don't find receptor tyrosine kinases in every, the most distant and simple animals on our planet. It's just super, super old school story. And then it's used over and over again. So in our bodies, the Wnt signaling pathway is used to put together all of our different organs. Because it's like a little tool, and you can use the same tool to put together different things, like that. So the same tool could be reused repeatedly, a screwdriver, to put together different parts of a car, or also not just the car, but also the lawnmower, and even the electric mixer, really different things. Fruit flies, and worms, and humans, and fish, and just all down the line, like that. So, you got, the authors you gotta put that in there, like that, okay? So that's an example of that one, frizzle, so unique receptors. Then here's another one that's just like it, massively significant in life on Earth of animals, not plants. Plants evolve different mechanisms to form multicellular plants than animals. And it's called the Notch receptor, and it's shown in our book. So here's the figure of the Notch receptor, and I can just add it. This one's pretty simple and pretty sweet, and it's just a curious one to see, because here's Notch, like that, and that's the receptor. So in the previous one, we were calling the pathway the Wnt pathway, because it's the signaling molecule. It's just the history of the naming of this. There are a couple of ligands for Notch. One of them is called delta. And, of course, they're important, but, people really worked on the receptor first. I talked about that. And here's the deal with Notch. When delta, or one of the other ligands, bind, what happens is there's a portion of Notch, see that? That gets cut off, that gets clipped off, and floats away, like that. And it's called the Notch intracellular domain. It's in the cytoplasm. So, and Notch into an ICD. See, it was part of it, and the enzyme cuts it is a protease, a proteolytic enzyme that cuts proteins, it cuts it in spot, and it just releases it. And NICD is a transcription factor. Is there any amplification in this signaling pathway? No. One receptor produces one transcription factor piece, like that. So it's a really curious receptor, like that. It's, I think, thought to be a really good switch, because you see how the transcription factor is not in the nucleus, right? It's in the cell membrane as part of a receptor. But when Notch gets activated, choosh, like that. And you have to keep making Notch, right? You have to keep making some new Notch, because that receptor is used up, like that, like that. So kind of a wacky one, like that. And there's plenty of wacky stories like that at this molecular cellular level in life on Earth, because it's just how evolution came up with a really cool thing. And once, yeah?
So what gets expressed when NICD binds to the DNA?
What kind of target genes? Yeah, so what's a target gene for Notch? And yeah, and I didn't list any target genes for Wnt or for Notch because they really are these tools where they can activate different genes depending on the cell, right? So like Notch is involved in also a variety of organ development in us. And it turns out that the different target genes are activated in different cells. And that's because there are other transcription factors that work with these guys. So when we were introducing it, we just would call those Notch target genes like we're doing. And remember, I've started doing that, right? So, you know, when we first got our first muscle differentiation videos, we're like, MyoD binds to myogenin, and then myogenin binds to muscle genes. And now we would say, well, MyoD binds to MyoD target genes. That includes myogenin, like that, because they almost always bind to multiple targets. And a lot of times we don't list them like that, you know? We talked about estrogen receptor. Oh, wow, there are lots of target genes for estrogen and the estrogen receptor. Like that, lots of target genes for FOXO. And sometimes we pick an example because it'll something we can really go with, you know, along the way, like that, you know. So we said myogenic because we could learn in detail about that. But it not can activate a lot of different target genes. So we would just say it activates target genes, like that, leading to all kinds of different cellular responses, yep, depending on the setting. Yeah?
Is CSL the same as TCL?
Is
CSL that it's binding to?
Oh, CSL? Yeah, that looks like it's a co-regular. I didn't really realize that it is. And I don't really know anything about CSL.
Okay.
Yeah, but Notch is the Notch intracellular domain is a transcription factor. Do I know that it's a co-regulator apparently based on that drawing? But
Okay, so
yeah.
it kind of acts the same as the
Wnt, yeah,
So it
yeah.
attaches to something.
I guess so. Yeah, I guess so. I just never paid attention to that.
Okay.
Yeah, but here's an example of Notch. So several students, Naomi is one of them, do these internships down at Maine Health Institute for Research. And it's got, you know, a big building, lots of scientists in it. And in the fall, the developmental biology lab class, we go down there on a field trip because there's a lot of developmental biology. You study Wnt, and Wnt's a big story in developmental biology like that, you know, how things form and grow. And one time, it's probably 15 years ago, we went on that tour down there, and one of the scientists said, in this great big, you know, biomedical research building where people are trying to make, you know, discoveries that really help human health in a whole range of ways, one of the scientists said, everybody in this building studies Notch. It's just like that. So it goes back to Naomi's question, which is, what does it activate? And the answer is it depends on the cell, depends on the time, because of that kind of biochemical context will be different. And so I don't even know if I know a specific, you know, target gene of Notch. Yeah. Yep. And so that's another one. And look, we get to add it to our transcription factor list, right? Because Notch gets cut and releases this NICD transcription factor. So really different story, right? Where the protein gets cut and now releases a transcription factor that's activated. And it was inactivated because it was outside of the nucleus, right? It's got to get into the nucleus. We know how FOXO gets blocked. It gets phosphorylated. This one can't get into the nucleus because it's part of a protein that's in the cell membrane, like that. Yep. Okay, so a nice story like that. And then the next one is called TGF beta receptors. If I could draw that up there so you can see that better. And that's the receptor. And this is a big gene family in us. In humans, just that, I don't know, you know, they're saying like maybe 40 human genes that are these TGF receptors, but they're all really similar. So it's considered a unique receptor that's distinct from these, because the other categories people don't really consider the G protein coupled receptors to be a bunch of paralogs, even though they probably are. They're just so distantly related that people would say, oh yeah, they're homologs, they're in a family, but they really don't dwell on that. Whereas with TGF-beta, they're like, yeah, that's just one TGF-beta receptor, that's just one big family. And just, again, these three, so Wnt signaling, Notch signaling, and this TGF-beta family, people don't generally call it, that's kind of the name of that gene family. There'll be specific names of receptors in there. We're not going to learn them, because again, these are massively famous stories in life, in animals, but let's just be realistic. So what do we want to get out of the TGF-beta receptor superfamily story? And that is that the TGF-beta receptor activates a small family of transcription factors. TGF-beta, I said, is the receptor, and people do call them the receptor because the receptor's official names are really lame. They're just called type 1 and type 2, and it's a dimer. And so TGF-beta receptors are members of the TGF-beta receptor big family of ligands like that. And they phosphorylate targets that are called SMADs, SMAD or SMADs, because it's a small gene family, and they phosphorylate them, and those become transcription factors. So with the TGF-beta receptor family, we get a simple story where we add to that one. Gets turned on by phosphorylation. And so I go through those fast because I want to emphasize, it'll be like maybe one question about Wnt signaling and one question, about Notch signaling and one question about TGF-beta signaling. And they might be about the that, or they might be where it's coming up in the setting up a list. Some transcription factors get regulated by phosphorylation because we have a nice list of those, right? You can pick, I'll say, list 3 examples of transcription factors that get phosphorylated to regulate their activity, and and what you know about them, like that. And so that's that. And I went through that kind of quickly because it is small packages. The other ones we were really kind of introducing principles of signal transduction and so forth. Okay, and so that's that. Okay. And so take a look at that, right? The figure is small, but there it is in the book, and there's a section about it. And I want to emphasize we really need to learn about that pathway. So, yeah. And so then I'll just show you that I'm not talking about, here's a totally other unique one that's in there that I'm not talking about, that you might want to learn something about, a little bit about. And it's this TNF receptor. And raise your hand if you took immunology. And then raise your hand if you took immunology and if you know what the TNF receptor is and NF-kappa beta. Raise your hand if you learned about that in immunology. Because, just as for example, why is this in there? So in our immune system, our immune cells are floating around and then they get activated. And there are a bunch of different kinds of immune system cells, and they get activated a bunch of different ways with a variety of different cytokines and stuff. And one really common switching mechanism is for the immune system cell to be bound by a TNF, and then that activates a cellular response that includes activation of new gene expression through this NF-kappa B. And so I didn't talk about that, but it could be something on an exam, but I know it's something that students that took immunology learned about, hopefully kind of a lot, and so I might not ask that, because that doesn't seem quite fair, like that, But I would just show it because it could, right? It's a different one, it's got its own section, and another really famous, unique receptor. But I just give you that example of there's stuff in the book that I didn't talk about, right? Because I don't want to overemphasize all these unique receptors and say, oh, that's the most important thing. No, the most important thing is the receptor tyrosine kinase and the G protein coupled receptor pathway, because we really dove into those. And we could dive into these too, but we're not. That doesn't mean we're not going to have any of those questions on the exam, because the book talks about them and they talk about it a good amount. There could be a little bit, a little bit. There won't be a lot of questions on the exam from the book that we never talked about. Would be, some like that. Okay, so that's that, like that. Okay? And so, let's see. Oh, that's just a zoom in on that kappa beta. And so it's a little bit out of place, but that just shows the flow. No, it's not out of place because it shows the flow of the chapter. Started with nuclear receptors, then they really wanted to talk about G protein coupled receptors, then receptor tyrosine kinase, then these receptors that they referred to as coupled to transcription factors because they all were, it turns out like that, but they do a lot of different stuff. And I just want to say a very little bit about the last section, which is this idea of regulation of the pathways. So this idea of amplification and integration. And remember, integration includes checks and balances and also crosstalk between these pathways. So lots of times it takes more than one pathway to get a cellular response going. And that's this integration, this crosstalk between totally different pathways, this check and balances. You know, like one pathway is like, let's go, but it's a really important thing. And of course, that's a big deal in cancer biology because cancer cells are usually cells that are growing out of control and they've lost some of the checks and balances like that, okay? So I just mentioned a little bit about that, and that is to, you know, I wouldn't try to memorize this. This is just trying to introduce you that pathway, you know, and look, see, it's showing that RAS and that MAP kinase pathway leading to cellular responses. So it's trying to give you a different kind of visual on a pathway and then make comparisons like that. And so see, this is that immune system pathway like that, important stuff on that, and some other, you know, totally other things we didn't talk about. And so it's, you know, it's kind of that last section of the chapter what's looking ahead, you know, where it's like, oh, you know, this was just a brief introduction to a lot more, like that. So it's some of that. And then here's just an example to see this. I mentioned this before. Here's a receptor tyrosine kinase that's activating both the, just those same receptors, activating both the RAS, RAF, MEK, ERK, ELK, MAP kinase pathway, and also the PI3 kinase pathway. So, you know, lots of flexibility too in these signal transduction pathways. Receptor tyrosine kinase might activate one of several, or it might activate two of them. Okay. Here's just this, just to show you this. This is that the PI3 kinase story, where that's that FOXO story. There's AKT, and we said, oh, AKT is a big deal. It's a big deal. It's a kinase, and here's what we saw. It phosphorylates transcription factors like FOXO. See, it's not just saying FOXO here. Depending on the cell and the timing, you can activate a variety of transcription factors. So really activate a variety of responses. And then this is just a little bit more. So it also says, it also does these non-genomic responses. So here it's saying metabolism, translation, like that. So other responses, not just transcription factor. And then here's just a little foreshadowing over this way. It's activating a different pathway. And so that pathway is foreshadowing. Remember I said that after the first exam, we'll read chapter 17, that's cell cycle. And then as we learn about that, we'll learn that in the cell cycle, yeah, cancers usually accelerate the cell cycle. But when a cell does that, there are checks and balances where chapter 18 says the cell should just go through apoptosis. It should go through programmed cell death and stop, you know, just end its life like that. And so that's why we want to go from chapter 17 cell cycle to chapter 18, which is about partly about programmed cell death. And that's this AKT doing that. Because just to kind of foreshadow, what we'll see is part of the checks and balance of these signal and transduction pathways is a lot of times when a cell receives a growth factor to say, go, like that, like hurry up and heal, you know, go through the cell cycle and heal the skin like that. The same cell at the same time often is getting from the same signal transduction pathway like an increase or decrease in its likelihood of going through programmed cell death. So, you know, just to kind of just glance at that, you know, our book is doing it, putting it there to show you a variety of things that have put that one there. So I would say that, you know, like, right, you're like, the cell gets a signal, yeah, you should go for it. and the cell gets worried. Am I gonna screw this up? If I start dividing really fast, is that, I don't know, I don't wanna do the wrong thing. I might just go through programmed cell death. And it's supposed to, really frequently, cells are just supposed to go, cause it's trying to prevent dysregulated growth leading to cancer and stuff like that. So just a little bit, in this last section about, taking kind of a bigger picture like that. And that's that. And I think we're done with, oh yeah, that's just, we won't talk about that. We can, if people have questions about that section, we can dive into it some. But that's that, okay? And so with that, I would say, I'm done talking with the kind of emphasis I want to give in the first round of what's in that chapter and what's in the book for our test. But, students can bring in questions like that. Sometimes a student will email me a really good, good question, clarification question, like maybe that one question about the notch intercellular domain, like at the weekend before the exam. And so sometimes I might send out an email or post a Brightspace thing will explain, this. So just be aware of that, we might have an announcement. Doesn't happen very often, but just be aware of that can happen. And then the other thing is sometimes I'll make a little video to be like, oh yeah, we gotta kind of dive into this. And that becomes new material like that. Anytime a student asks a question in class, or if it's one I wanna share with everybody, that's, material I gotta do. Yes, Sophie.
Should we know the mTOR pathway?
No, the mTOR is intracellular protein, cytoplasmic protein, and it comes up a couple of times. There's one version of it that's in the pathway toward activating AKT, that PIP3 in the cell membrane activates that, and then activates AKT. And I said I wouldn't worry about that too much because it's actually a multi-subunit complex. And there's also a different alternative pathway to get AKT activated by binding to, phosphorylated PIP3. So it's not, that TOR is not a big name. It has a kind of a variety of sort of settings inside inside the cytoplasm as a part of the signal transduction pathways in a cell, and it's actually like kind of as famous as FOXO in regulating kind of growth stories, and so the book talks about it some, but I would say I wouldn't dive into details of TOR, or they call it mTOR also. Yeah? Yeah?
You would have been drawings to the exam?
Yeah, totally. So I'm left-handed, you see, I'm not an artist, but totally will have to draw and label stuff on the exam. And there'll be a thing where if I say, you want to just follow the words, right, in the question, because I might say, draw and label the, PDF receptor pathway. You got to draw and label the whole thing for sure. And other times I'll say, draw and label it, do that, and then briefly explain it, right? So make sure you look at the words. And, the questions are very direct. And if I don't say draw and label it, if I say describe it, you're super welcome to add drawings. But also if I say, briefly explain it, you also have to have some words. But in any time I say, explain this, explain that, you can include label drawings. Sometimes if it says draw and label, that's all I want, I want that, right? Other times I'm not saying draw and label it, but you can, but when I'm saying like explain it, describe it, also use words, right? Use sentences to explain what's going on, not just, I drew it, that should be good, right? Yeah, but there's tons of possible drawings, and not just like pathways, but also like methods, right? We learned those methods and regulating transcription, learning about transcription factors. Some of those would be very nice to demonstrate, what's going on with fewer words, because words are often, worth 1,000 words. A drawing is worth 1,000 words like that, pictures worth 1,000 words. So it can be super helpful to demonstrate, what the ideas are. So lots of times you would do a kind of combination of drawing and describing. Yep. Yeah, for sure. Yep. And again, not judging your artistic skills. Remember, I've emphasized draw big, like that. That's also part of demonstrating that you know something. And so my impression is that when a student doesn't know something very well, they draw it really little. You know, I think they're just kind of shy and hoping that it looks good. But if I can't figure out, you know, if it's really little and stuff like that, so just make a big drawing, you know, and it's just a simple line drawing. It's not like art, you know, with shading or anything like that. It's just a label, you know, simple label drawing with arrows and, you know, cell membrane, and label it, you know, cell membrane, nucleus and stuff like that. And all of it is just demonstrating, you know, like that. I like drawings because it's a good way to learn it. It's a good way to remember it. And it's very easy to grade them, you know, because I'm like, that looks good, you know, overall, great, you know, that's, you know, there might be a little detail in it, but I'm like, looks good to me, yep. Yeah, when I grade stuff on this, you know, sometimes it's, you know, they'll get down to saying, well, you got half a point, right? But, you know, it's, you know, I'd like it to just be, that looks great, 2 points, you know, or 1 or none, you know, and, you know, and not like 1.5, you know. But the way I write questions, sometimes it'll be like 2 points to list 4 things, and I'm like half point for each of those to get that many. Yep. Other questions people have? So here's some more stuff that'll be on the first exam. It's more stuff about MYC. So remember, MYC is a transcription factor. It's a big time oncogene. And what goes on with MYC is if you just make too much normal MYC, you're in trouble. So that, remember, we said, oh, there's really 2 big oncogenes from the 1970s from those oncogenic retroviruses we're going to have on our exam, and one is RAS and one is MYC. And they're very good examples because RAS, when it causes trouble, has mutations in the coding region, in the transcription unit, in the product. And MYC, when it causes trouble, has mutations in the cis-regulatory region. So we're going to just dive into that more, okay? Some more on that, okay? And so this is one of these examples where I've highlighted one of the sentences in this little phrase, you know, and it's just not, I think if we could dim the front of the lights or something like that, but I'll read the highlighted sentence out of this, you know, just so you hear, because, you know, I could just say it, but I'm like, it's from a review. So it says, the MYC, which is kind of funny, the MYC is a global transcription factor contributing to various cellular processes, one of which is hematopoiesis. And hematopoiesis is the constant formation of new blood cells from our bone marrow. So in our bone marrow, there are stem cells that are constantly dividing. And you know, people have bone marrow transplants and stuff like that. There's a really important part of our body. In the bone marrow, these stem cells are constantly dividing, I don't know, millions and millions of new cells every day. And they make all the blood cells, so the white blood cells of the immune system and the red blood cells carrying oxygen. So that's hematopoiesis, like that. And so what they're saying is that's a big role for MYC. Where? In the bone marrow. Stimulating that massive proliferation, right? And we and other vertebrates all do that. We're going to make new blood cells all the time throughout our lives. Is that dangerous? Yeah, right? You're playing with fire when you're just like, we're going to make a lot of cells in that bone marrow, right? And there's some cancers that are blood cancers, like myelomas and leukemias and lymphomas, that involve dysregulation, disruption in this, and some of them specifically involve MYC, okay? So we're just setting that up. You know, we're going to bring our thoughts to that hematopoiesis in the bone marrow that's constantly happening. And we've already talked a little bit about this. Remember, we said, you know, RAS is commonly mutated in cancers where it's in a location that the RAS is normally involved in regulating growth. Like in intestinal type cancers, they very, very often have mutations in RAS because RAS is normally involved in the constant replenishment of the cells along the lining of our intestine. Our intestines, like our outer body skin, you know, the cells die, they get rubbed off like that. We gotta replenish them all the time, like that. So then we talked about PGF. Well, that's, you know, it's got roles in other places, but, you know, platelet-derived growth factor, right? You know, wound healing in the skin. So there's some skin cancers like that. Not all skin cancers, but certain specific ones have problems with the PGF receptor signaling pathway. So we just are setting it up to say, uh-oh, what's the kind of cancer that MIC could be involved in? And we won't be surprised that MIC gets involved with some of these blood cancers. Not all of them, but some of them, because the setup is just there. You know, the biology is just there for us to be like, yeah, phew, you know, constantly producing lots of new cells, right? Like that. So just to reread that, because I stopped. The MIC is a global transcription factor contributing to various cellular processes, one of which is hematopoiesis. Oh, here's a number. In the bone marrow of adults, 300 million cells are produced every minute, right? So that's just like a little fire, you know, and cancer's like a big fire like that, you know? So, and MIC is just like, let's go. Yeah, MIC is rowdy, like that, okay? Next one, highlighted here. The RAS-RAF signaling cascade has an important role in the regulation of the MIC, and they say promoter. So let me just mention that. So I've said before that you know, definition of a gene is it's physical, it's made of DNA, and it's composed of these two parts. And we definitely know there are these two parts, and one part is going to make the product. So that's got to make the RNA, like that. And then the other part is going to regulate where, when, and how much, right? And that's just true. How much we've said there is just super true for every type of life on our planet. And I mentioned that people use some somewhat different terminology. And for a student, you know, oh, darn it, you know, that's frustrating. And we use this terminology because it's great. So that's the transcription unit, right? It's great because that just means it's the part that gets transcribed, folks. And then this is best called the cis regulatory region. And we've got like enhancers here, but we also learned that not everyone's called those enhancers. Sometimes they're activators, repressors, so we call them response element. The estrogen response element can be both a repressor and an activator. Yeah, not a good name, enhancer. Then promoter, we say the promoter is always right next to the start. That's where tau binding protein, TBP, and the pre-initiation complex assemble. So you can move that enhancer around. You cannot move the promoter around because it's literally, it's like at an airport where you have some big spotlights that show the airplanes where the airport is, and then you have some little spotlights right at the end of the runway and say, put your plane down right there. It's literally that. We got some big enhancers to pull in the RNA polymerase and the other regulatory steps, and then this is just like, okay, this is where it's going to happen, like that, okay? So it turns out that in certain settings, and we don't need to answer this on an exam, but just to be aware of it, and also be aware of, in other classes, past or future, or on the web, like that, you might hear like a different term, and you'd be like, wow, is Dave Champlin really that stupid? And the answer is no. No, he's even dumber than that, right? Yeah, like that. But because it turns out that some people in some settings, they just call the whole cis regulatory element the promoter. Are we going to do that in our course? No. If you did it, it'd be wrong. But here in this science review, they talk about the MIC promoter, and they just mean the whole cis regulatory region. Part of that's because it's some of that goes for just historical context, People didn't always understand it, and so they just called it, the promoter like that. So don't worry about it, but since, it says here, I just want to explain it. But if you say promoter for that whole region on our course, it's wrong, like that. The people that call it the promoter actually have kind of a legit reason, because they call the promoter the proximal promoter, which means nearby, and they call the enhancer the distal promoter. And they're not really like married to those terms. They just are like old folks. I'm kind of like even older than me. So just don't worry about it. You know, it just be in these different settings. So again, the RAS-RAF signaling cascade has an important role in the regulation of the MYC-cis regulatory region. Small GTP binding protein RAS promotes MYC expression by inducing the RAF-MAC kinase MEC pathway. Platelet-derived growth factor receptors and SRC kinase, we didn't learn about that, also augment the activity of RAS proto-oncogene, which results in activating the mitogen-activated protein kinase pathway. So, you know, we learned a lot of those words. We could kind of navigate through there like that. Okay, so they're talking about MYC, and we've already learned that. We knew this, which results in activating the mitogen-activated protein kinase pathway. However, both PDS receptors and SRC mediate the induction of MYC expression independently of RAS. So they're just saying it's going to be one of these. You got to have two signals because, you know, it's like, you know, MYC is a gun, and you better have, like, take the bullets out, put the safety on, lock it in the cabinet, hide the key to the gun cabinet. You have to have multiple checks and balances, and they're just saying that using a bunch of words like that, okay? So they say that. So we show this again. Oh, we know what they're talking about, like that. And then it says, uncontrolled MYC expression is observed in human leukemias and lymphomas. So we knew that, right? We were learning about that. Oh, and then it didn't highlight this, but we just read on. Generally, MYC overexpression does not stem from point mutations in the gene. Rather, hematological malignancies such as acute lymphoblastic leukemia, chronic lymphocytic leukemia, and myeloid neoplasms, overexpression is mainly due to, and then it says some examples, and all the examples are changes in the cis regulatory region, okay? So that's what's going on. Then it says the MYC expression pattern is tightly regulated in normal conditions, though MYC is often dysregulated in cancers. Okay, another statement here, MYC expression is highly controlled at several levels, okay? So we're gonna learn about that. So MYC, and see how I didn't go all the way to start over, but we just need some room because MYC is gonna be regulated. MYC is a transcription factor that goes on our list, and it's just in its own category. Because one way it's regulated is transcription elongation. So that means MYC, the MYC gene, has an arrested polymerase. That's common. That's a rate limiting step. To activate it, you gotta get that arrested polymerase. But if you look at MYC, MYC has multiple spots all the way down the transcription unit that cause the RNA polymerase to stop. So it just does not want to get made. So this transcription factor is regulated partly by just an intense transcription elongation. That means the RNA polymerase just is struggling to get down the gene because the gene is like, MYC is like, do not make me. You know, don't mess up because this is in some trouble. Okay. So, and then it goes, it's listing them here too. mRNA stability, so that's it. It gets transcribed and the RNA is super unstable. So the RNA would be mRNA and it's got to get processed and go out in the cytoplasm and it just gets destroyed really fast, like in 5 or 10 minutes. So it's another example of regulating a transcription factor and it's MYC, and these are all just don't make it. Don't transcribe the RNA, don't have the mRNA around, so we make the protein. Don't do this, right? So then here's another one, translation. Ribosomes don't want to translate the mRNA. They just don't. They're just like, yeah, no. I can smell trouble, you know, right? Like that, you know, you're playing over a friend's house when you're 12 and they're like, my parents have guns in a locked cabinet and I know where the key is. And you're just like, no. I think my mom's calling me for lunch, you know. It's just like that, and it's just wicked, right? And even after it gets made, it's not stable. The MYC protein just gets destroyed. So this is just an example, right, where you're like, MYC by itself is just like, and they're all turn it off, stop it, block it, like that. So can't transcribe it, can't keep the mRNA around, can't translate it, can't keep the MYC protein around, right? Okay, And you might already be able to guess what's going to be the problem. The problem's going to be in the cis regulatory region, and it's just going to be make way too much. Overwhelm all of those blocks, you know? Just make tons of it, like that. Tons of the RNA, tons of the mRNA, make the ribosomes make it, you know, so that whatever, you know, block is, it just goes over too much like that. So it's really a sad, sad thing. Generally, MYC overexpression does not stem from point mutations in the gene. Instead, there are big changes in the cis regulatory region because it's just like, let's go, right? And it just makes it too much, right? So we brought up this before. How much? Too much by a lot, right? Where, when, how much? The changes in the cis regulatory region are all going to be how much, not where, when. It's already in the bone marrow like that, can get made and used. And so we're going to learn 3 examples of overexpression of MYC. And the first one is one we already know. We talked about it. Retroviruses have these really strong cis regulatory regions because retroviruses are virus, and they're like, when they infect, they're like, make me lots, you know? I'm just going to make a lot of virus, infect this cell, and get, you know, lots of copies. And when MYC got connected to those, right, too much MYC, and it just overwhelms all those downstream regulatory steps. Just too much MYC like that, right? Okay? And we'll go through them like 3, one at a time. But that first one, we know about that. And then these others are going to be cancer stories. Not the virus, right? Because it was like, you know, people studied the virus in the 70s and then they moved on to real bodies, real mice, and real human diseases, okay? So archogenic retrovirus, right? So back to this, you could draw this. There's the genome. That could be MYC. Actually picked up MYC. What part of MYC? The transcription unit. And it's wild type, super normal, regular MYC. And they just have really strong cis regulatory region. When the virus infects, and you know, it gets copied into DNA, the DNA gets inserted, you just get really strong expression of, unfortunately, the host gene MYC. Any problems with the MYC protein? No, it's fine, but it's just too much of it by a lot, okay? And that's the full first example of the 3 examples. And again, that goes back to the oncogenic retrovirus story, right? That's they got hold of it. But then they found comparable examples that didn't involve a virus, something in our bodies, you know, that would lead to a mutation in MYC that would be making too much normal MYC. So the next one is called a chromosomal translocation. So remember, we can just draw this, right? Like we can draw this. Remember how we can draw pictures of where we know how to misexpress the GFP, the jellyfish glowing gene. We can misexpress that in muscles because we would take 2 genes, right? And we take the GFP gene from jellyfish, and then we would take the cis regulatory region from mouse myogenin, and we would intentionally join that part with that part, create that trans gene, and make a transgenic animal. And so we know all about that, right? When you swap the cis regulatory region, you put the transcription unit under the control of a different cis regulatory region. And it'll just do whatever that is. So that's in a test tube, right? So it turns out there's a kind of mutation, and I'll draw it over here, that goes like this. It's a class of types of mutations called a chromosomal translocation. And what it looks like is this. And so we use, you know, examples of humans. So in humans, we have, you know, 23 chromosomes like that, or 22, and, you know, a sex chromosome like that. And they're named based on their length. So there's a chromosome that's pretty long, and that's like chromosome 3. And then we'll draw a different chromosome, and this is just, you know, made up to kind of demonstrate this idea of chromosomal translocation. And that could be chromosome 11. I don't know why. I don't need to Roman numerals. That's chromosome 11. That's chromosome 3. Okay? Like that. And they're different lengths, and they have different genes. Okay? So there's a kind of mutation that where a chromosome can just get torn, and the body's going to try to repair it. They're going to try to glue it together like that. and do a DNA repair thing and fix that error. Okay? And the way to think about it, I'd say, is to think about it like one of your textbooks, right? And you're walking down the stairs and you drop your textbook. Oh, I already did this intentionally with mine, right? I just cut my book in half, like that. So it just gets torn, and you can just glue it back together. And it got torn on a certain page, randomly, and that page couldn't be a gene. So when we say, oh, there's been a chromosomal break, it could be in a gene. And then, rarely, right, rarely, there could be two chromosomes that get torn, and in repairing it, the cell can make a mistake. And you drop your biology book and your math book, and they break in half. You go home and you glue them, and you accidentally glue the front half of your biology book with the back half of your math book, and vice versa. And that's a chromosomal translocation. And you're just like, dang. Because what's going to happen is we're going to tear a gene in half on chromosome 11 randomly, and one in half on chromosome 3. We're going to glue them together. And it's like saying we're going to glue accidentally your biology book together on some page where the next page is a math page. And that's going to make a weird combination like that. So that's what's going to happen with a chromosomal translocation. And, scientists could see these under a microscope in patients' cells. Even in the 1950s, they could see these chromosomal translocations, and they could say, like, it's a chromosomal 3 to 11 translocation. And even in the 1950s, there were specific types of cancer that had that chromosomal translocation. That's how they diagnosed it. They're like, this person has a kind of blood cancer, leukemia, myeloma, lymphoma, like that type of thing, and we're going to give it a specific name because everybody that has that translocation has cancer. Okay? And so these chromosomes have specific genes on them. And there's a chromosome that has MYC, and MYC is one of those. They saw it a long time ago. They had no idea what gene it was, right? So we're going to see that. That's our next example. And what it's like is the same idea that we did where we make we have to stop. We make transgenes in a test tube, and this is not a transgene. It's a total random thing. Accident happened, where chromosomes break and they get glued together wrong. And just to, you can get up and pack, but just to kind of complete the drawing, what we end up with is, a chromosome that goes like, it's partly chromosome 3 and partly chromosome 11, because they just get glued together like that, where the two parts get glued together. And then there's, they're stuck that way, right? Like that, they're fixed, and the body goes through and copies them and stuff like that. Okay, we gotta stop. And that's that, okay?